Great. I'm also going to time myself, make sure I don't go over. Hi, it's nice to meet you all. My name is Charlie Hoyt. I'm from the Laboratory of Systems Pharmacology at Harvard Medical School. And I'm going to give a presentation on the biopragmatic stack, which I've changed the title. It's now Biomedical and Chemical Semantics for Humans, because this is a generalized system for, I think, a lot of things in the life sciences. And yeah, it does piggyback really well on a lot of the discussions we've had today, especially about ontologies versus controlled vocabularies and kind of what are those things. So I put a link to the slides in the chat. Feel free to follow along, download it, do whatever you want with it. Yeah, so some of the big questions I think we have to ask about the semantic spaces in the biomedical and chemical domains is like, I think we probably are past this already. Why do we need stable identifiers for biomedical and chemical entities? I think this is pretty clear that we need to have some sort of system for referring to things that we can control. But like the next big question is kind of like, who comes up with these nomenclatures and these controlled vocabularies? And like, who keeps track of all of those lists? Like, for example, if I have a domain I'm interested in, how do I find the ontology or controlled vocabulary that's right for me? And then another big question is like, how do we know when two things are the same? Both in like the physical world and also in the semantics world, once we start using an ontology or controlled vocabulary to talk about a thing. If we use two different controlled vocabularies, they may have overlapping domains. So the same thing may pop up in two different vocabularies. And then, once we know what we're talking about, how do we get at the data about chemical entities in sort of a general sense? So I'm going to present a couple different tools and a couple different ideas and some code and some data that we put together in my group to solve some of these questions. I'm going to start with the we're going to have to start talking about how identifiers actually look in the semantic web. And if you're familiar with RDF or Sparkle or triple stores, you've probably used URIs to talk about things and as stable identifiers. So that's kind of what we're going to do is we're going to talk about this chemical alcyrpolone. And on the top, we have a URI that sort of refers to this chemical. And this is the first party URI. So this is part of the Kibi website. And it's talking about this Kibi molecule 138488, which is the local unique identifier for alcyrpolone within the Kibi namespace. So you can see that there's kind of this prefix part where every Kibi molecule can be resolved by building a URL that has this prefix, and then you use the local identifier that comes after it. So there's alternative kind of shorter syntax for semantic web identifiers like URIs, which is called compact URIs or queries. And this sort of abstracts away this kind of garbage looking URI prefix, and it gives it something more meaningful, Kibi. So if you're familiar with Kibi, this nomenclature and ontology for chemicals, you know that like a query that starts with Kibi refers to something that comes out of this URI, and it refers to alcyrpolone. The question is like, what are all the prefixes? So sorry to jump back. So Kibi is now like the prefix that goes with the database and the nomenclature, which is from the chemical entities of biological interest. And so people typically know what the prefix is that goes with Kibi, but sort of the more general question is like, what are all of the prefixes and the metadata that goes with them? So like the URI prefix string, knowing what is the like kind of long name of the resource, who is taking care of it, who's responsible for it. And so yeah, a big question is, what are all the prefixes? And so what you want to do is look into sort of what's called a registry, which lists prefixes and their metadata. It turns out there's actually a lot of registries. One common one is Prefix Commons, identifiers.org, perhaps you've heard of those. All of them seem to have a little bit of a different domain focus. So another big question is like, are the prefixes in one registry the same as the others? Are there synonyms? Are there collisions? And this is actually not a very easy question to answer directly. And then another difficult question is, how good is the coverage of each of these registries? It's quite varied. And can we do better than what exists already? And the answer is yes. And so the bioregistry is sort of this meta registry for the life sciences, for chemical and biomedical information. And it maps between all the prefixes and the metadata between different places. And it helps standardize your data. So if you're using queries, it makes sure that you're using the right prefix, and it helps us standardize. All right, so next step on top of that is like, where do nomenclatures come from? We talked about ontologies, but there's also other kinds of controlled vocabularies like the Entregene database, Interpro, and then other like resources that are actually databases like the Gene Expression Omnibus. Now, ontologies are using this wonderful standardized format like OWL and the OBO sort of serialization of it. And it makes it really easy to traverse ontologies. But why don't databases like DrugBank and Chembl use some kind of standardized format for giving information about their chemicals? this is a problem. So Pyrobo is a tool that does a couple things. It helps pre-process ontologies, but it also helps convert databases like DrugBank and Chembl into the ontology format. So we can sort of aggregate the names, the synonyms, and all the properties, including cross-references out of these ontologies and databases at a sort of scale, and sort of provide unified access, like in this example, to getting the name for something given its prefix and its identifier. So moving a little bit forwards, one of the things that really interests me is mappings between different resources. And we talked about this earlier as well. So first of all, we need to standardize meta nomenclature. What I mean by that is like depending on when you're doing data integration, if you have two different data resources, they may use different prefixes to talk about the same thing. So we need to standardize our prefixes. And then we need to prioritize our nomenclature. So if we have a drug bank query and a Chembl query, both referring to the same thing, we have to decide which query we want to standardize to so we don't have duplicates. And so we can basically use string operations to check for things being equivalent. So on top of this super aggregated ontology database, as I said, I could build with PyObo in the last slide. I took the whole OBO foundry, the Open Biomedical Ontologies Foundry, which Chris Mungle is going to talk about very briefly in the next talk as well. And I took all of the cross-references out of all the ontologies in PyObo. It turns out there's about 20 million of them. And yeah, you can use these with some quite simple algorithms, either in memory or in a graph database, to implement one of these prioritization algorithms. And so all the code that does this is inside PyObo. You probably see at the bottom of the slides, I'm linking to blog posts I've written or code that goes with a lot of these things. So feel free to follow up after or ask questions later. So yeah, I mentioned this is called the Inspector Javert's XREF database because Inspector Javert, he was the character from one of my favorite plays, Les Miserables, and he had this problem where he was looking for the main character, Jean Valjean, except this guy, his name kept changing. And so Javert kept following him from place to place where there was a different controlled vocabulary for all the people. And he didn't have mappings between his controlled vocabularies. So he could have used a cross-reference database. But there's one big problem with ontologies and databases and pulling all of these cross-references out of them. There's actually some systematic gaps in what's available. And so this brings us to the next kind of part of the biopragmatic stack, which is biomappings. And this is our way of addressing these systematically missing CREFs. Yes, a CREF, cross-reference. It should be XREF. So I'll give an example about cells, but this is also applicable for chemicals, for example. So we had a very specific use case where we had data from the DET map and the cancer cell line encyclopedia, which are these two on the bottom left. And we wanted to map them to cell ontology, but it turns out that none of these resources are like directly mapping to cell ontology. And the arrows are showing which resources map to other resources. So we basically had to go around the cycle to get mappings from CCLE through DETMap, through Cellosaurus, through the experimental factor ontology, through Brenda, and then to the cell ontology. This is pretty inconvenient, but you can make inferences about cross-references in certain cases. However, even when doing this, there's still incomplete mappings. So we had to go about generating these mappings and curating them. So biomappings is sort of an assessment of this, but also an actual workflow for retrieving all these ontologies or resources that give nomenclatures, making some predictions, using a couple of different tools like Gilda or PyObo actually has something for doing this I mentioned before. So building lexical mappings, then filtering down to the scope of like what you actually want to do curation on, helping you do the curation and then put the results back in to make sure that we can keep making new mappings and maybe improving these systems for generating the mappings. So yeah, there's on the scale of, maybe I think on the scale of 5 to 10,000 manually curated mappings, and there's like more than 30,000 predicted mappings. And at the same time, we're curating negative mappings as well. So when we find sort of non-trivial things that look like they should map together in a prediction, but they're not really the same, we curate those as well to avoid what we're calling zombie mappings. So we don't have to keep recurating the same negative results. So yeah, this is moving forwards. Now we have sort of a way of filling in these systematic gaps in the ontologies. And by the way, doing this in sort of an open data, open code scenario, so we can contribute these mappings back to the original resources. And then the last thing to talk about is the BioLookup service, which is about chemical entity information as a service. So once you're able to build this kind of unified aggregated database of all the names, the synonyms, the cross-references, properties, relationships between entities coming out of ontologies and databases that have now been serialized using this ontology format. You can sort of build a service that lets you look up all of these things. Now, when I mentioned PyObo, I gave this code example on the bottom where you can look up the name of something given its prefix and its local unique identifier. The problem with doing this is it's a bit hardware intensive. Key B is quite small, like less than 100,000 terms in it, so you can load that up no problem in memory. But when you want to scale that up to PubChem, which has on the scale of 100 million chemicals, it starts becoming a problem. And then there's other resources too on top of that. So you want to have all of those loaded up in memory. It's not really appropriate to spin up every time you do an analysis on your laptop. Yeah, so the alternative to that is kind of to have external APIs that you can hit. So let's say you want to use the PubChem API. You actually have this problem with rate limits. And turns out the PubChem API is a little bit unstable and kind of finicky. So if you're doing a lot of operations on it, sometimes it just doesn't return results. Yeah, and so we don't have any way of like solving this rate limiting problem or scalability problem with PubChem without sort of reproducing the service. So yeah, and then the last problem is, PyObo is written in Python. Not everybody's using Python. And so what we need is sort of a consistent language agnostic way to access databases or access, all of this aggregated information. Either it's like a database or a web service that doesn't have this issue with rate limits, doesn't have a problem with scalability, so you can spin up like new instances of the service to meet the user's needs, usually ourselves being the users. So the BioLookup service kind of hits all of these points. And basically I take this aggregated database I talked about with PyObo and distribute it in a prepackaged Postgres image in Docker. So you can just kind of pull the Docker image and run this database. And then there's also kind of a Docker image that has a thin API that wraps this database and making queries to it. So right now the BioLookup service is sort of running at this instance. It's got a couple of things that needs to be improved, like it doesn't have HTTPS at the moment. And it also is a couple of things that I want to add. I want to make sure that it can have synonyms and cross-references and relationships. And for like chemistry specific stuff, you can also pull these properties. A lot of molecules have like a SMILE string or an INCHI string that go with it to show the structures of these molecules. When you look at either the user page, which is kind of what you look at as a person, or the API, which is something you can embed into your applications. So this is the last slide. I know I went quite fast through all of these, but higher on the pyramid of the biopragmatic stack is thinking about information extraction, which is like, how can we allow people to write text that's interesting to them and not have to worry about identifiers or queries, any of this stuff, and build either NER system, named entity recognition, or just named entity normalization systems. So when you have these supersized databases of names and synonyms, it's pretty obvious how you can start to make your own systems or maybe train your own systems to do this task. Now, one thing to keep in mind is there's a trade-off between generalization and performance when you build these named entity recognition and normalization systems. So one thing that you can do to deal with that is to kind of do like context disambiguation. And so I'll give a shameless self-promotion to the Gilda system, which uses ADEFT to do this sort of disambiguation based on the text that surrounds the text you actually want to ground. And built on top of that is doing more information representation and information like relationship extraction. And so people want to sort of structure the relationships they find between biomedical entities and chemical entities in the text using things like biological expression language, biopacks, and SPGN. And so doing all the things that we mentioned before allows people to curate these resources, these sort of information representations with high quality, stable identifiers, but then look up the information that goes with those entities when it's time to show it to users. And yeah, in the end, a lot of these things support my team's sort of larger scale reading and assembly pipeline, which is called Indra. And if you want to know more about that, you can feel free to check out our team's website, but that's not really what this talk is about. But that's why I've been motivated to care about a lot of this stuff and how I've really gone deep down the rabbit hole and ended up working on some of these problems. Yeah, so I want to acknowledge all the people on the team that have built some of the things that I presented. Albert Steppi, Ben Giori, who's in charge, Diana, John, Klaus, Patrick, and Sam. Yeah, and we are funded by DARPA, so it's good to mention them. Yeah, so that's the last slide. Be happy to take any questions. I tried my best to stay on time, but that was
perfect. Thanks, Charles. Yeah, there's 2 questions in the chat already, so maybe we'll just leave it at those ones for now, so we can try and catch up a bit. So the first was from Bob about a particular piece of your diagram on slide 7.
All right.
The significance of the one-way mapping between the cell ontology and Brenda.
The cell ontology, when you download the cell ontology, it maps, has cross-references to Brenda, but if you download the Brenda system, it doesn't have mappings to the cell ontology. So that's what the directionality of those arrows means. You can notice between DepMap and Cellosaurus, which is the one on the top left, both of those resources have mappings to each other. So it gets two different directions. The second question was from Peter.
That doesn't slow you down. You don't care. As long as you have one way, you can do everything you need to do, right?
There's a big philosophical discussion to have about whether cross-references are one-way or two-way, and it really depends on the semantics of your cross-references. One of the really big issues is that cross-referencing isn't really semantically defined, like an exact match relationship. From Scoss is a really more powerful one. There's this OWL idea of equivalent class and equivalent to. And I think if you want to know more about that, you should check out the recent SSSOM paper, which talks about more of the philosophy of mappings and how we should be dealing with this stuff.
Great, thanks. And then maybe while Chris is getting ready and sharing his screen, there was a question as well from Peter about whether you have any interaction with Wikidata.
Yeah, so I think, well, it depends where you want to ask. So the bioregistry actually is pulling a lot of properties from Wikidata and considering properties as their own sort of database of prefixes and associated metadata. So there's mappings between the bioregistry and Wikidata. There's also in some places mappings between the entries in Wikidata for the databases or the nomenclatures themselves, which is a little bit different into the semantics that it sort of implies than the properties themselves, but they're quite useful to know about. The mappings would be potentially useful to put on Wikidata, and I've until now been a little bit scared of the Wikidata like bulk uploading process. So if anybody knows how to do that and wants to help and join as a co-author on the publication for biomappings, I would love to have you. So get in touch with me, please.